Hello everyone. I warm welcome to all participants joining us from across India and beyond. We the students are truly excited to present this webinar crafted with passion and purpose. On behalf of biotechnology students at the Center for Scientific Research and Development, Pupils University, Bhopal, I am delighted to welcome you the third session of the BioConnect webinar series, a unique by the student for the student initiative. My name is Somra, and I am proud to tell you that I am a student of MSc Biotechnology and a member of this webinar organizing committee to serve as the MC for today's session. We are discussing that this is students-led webinar on biotechnology for a sustainable future. Innovation in bioplastic production. As the world faces growing environmental challenges, biotechnology efforts powerful tools to help us build a greener, cleaner tomorrow. One such innovation is bioplastic production, a promising solution that reduce our reliance on fossil fuel. Today's session brings together innovation ideas and a shared vision to explore how science can lead us toward a more sustainable future. We extend a heartfelt welcome to all our participants and esteemed faculties members from us various institutions. Your presence adds immense value to this gathering and inspire us to committee nurturing such a vibrant academic committee. A very special welcome to Honorable Dr. Nirja Malik, Dr. Sudesh Kumar, our distinguished speaker, expert in biological science, environmental microbiology, environmentally friendly product and technology at University since Malaysia. Professor Dr. Subhash Jibore, Head of the Center for Scientific Research and Development at People's University, and our mentor. Ms. Priyanshi Sharma, Chairperson of the Organizing Committee of this program. This formally inaugurate today's session. It is my honor to invite our Honorable Vice Chancellor, Professor Dr. Nirja Malik, to deliver the opening remark. Ma'am, please proceed.
Thank you, Ms. Umra. It is not Honorable Vice Chancellor is busy in some other meeting, so I'll be addressing the gathering today. Good afternoon to all distinguished participants, faculty members, dear students, and our esteemed guest speaker. It gives me immense pleasure to welcome you all to today's special edition of the BioConnect webinar series, organized under the visionary leadership of students at the Center of Scientific Research and Development, People's University. This initiative, built on the empowering theme of for the students, by the students, exemplifies the spirit of academic curiosity, innovation, and leadership we proudly nurture in our university. Today's topic, biotechnology for a sustainable future, innovations in bioplastic production, is not only highly relevant, but critical to the global sustainability discourse. As we confront the challenges of plastic pollution and environmental degradation, bioplastics offer a promising science-driven path forward. We are truly honored to have with us Professor Dr. K. Sudesh Kumar from Universiti Sains, Malaysia. Welcome, sir, a renowned authority in the field of sustainable biotechnology. His groundbreaking work on bioplastics is globally acknowledged, and I'm sure that today's session will be deeply enlightening and thought-provoking. I take this sincere opportunity to sincerely appreciate and congratulate our organizing student team at the Center for Scientific Research and Development for their enthusiasm, professionalism, and vision. I also acknowledge the valuable mentorship of Professor Dr. Subhash Bore, Professor and Head in Center for Scientific Research and Development, and Student Advisor for gathering this meaningful initiative. To all our attendees, whether students, researchers, or educators, thank you for joining us. I encourage you to engage actively and forward the message of science for sustainability. With these thoughts, I am pleased to declare the webinar open. Let us learn, reflect, and innovate together for a cleaner and greener future. Thank you and best wishes for a successful session. Over to you, Ms. Umbra. Thank you so much.
Thank you, ma'am, for your inspiring word. We highly appreciate your support to students-led innovation at People's University, Bhopal. Now, I
Thank you so
invite the
much.
chairperson of the organizing committee to say a few words. Ms. Priyanshi Sharma, over to you.
Thank you, Umra. Respected dignitaries, esteemed guests, faculty members, and dear fellow students. Good afternoon. It has been an honor to serve as the chairperson of the organizing committee. On behalf of the entire team, I extend a warm welcome to each one of you to this important and timely webinar titled Biotechnology for Sustainable Future Innovation in Bioplastic Production. As we are all aware, faces two pressing challenges: the ever is of plastic pollution and the urgent demand for sustainable alternatives. Biotechnology gives us a beacon of hope, and innovation is the driving force behind that hope. By harnessing biological microbes and enzymes, scientists have developed the ability to produce eco-friendly bioplastics, materials that are biodegradable and significantly reduces our reliances on petroleum-based plastics. What's truly exciting is that this bioplastic from renewable resources. Bioplastic can be made from renewable resources like agriculture waste, starch, and even algae. With ongoing research and industries collaboration, we are inching closer to replacing the conventional plastic across sectors such as packaging, agriculture, healthcare, and industry. Let us remember scientific thoughts and alone are not only enough. Real changes demands awareness, progressive policies, and active cooperation between academia, industry, and government. Plastic become truly sustainable, accessible, and impactful. To my fellow students, I want to specially emphasize your role. The future of sustainable biotechnologies lies with you. Be curious, be bold. Question, experiment, and let work together to that not only advance science but also serve the planet. With this thought, I thank you once again for your presence and participation. I look forward for an inspiring session filled with innovative ideas and meaningful discussions. Thank you. Umra, over to you.
Thank you so much, Ms. Priyanshi Sharma, for your welcoming words. Distinguished guests and fellow students to take the session to forward. I now hand it over our moderator, Sandhya Yadav. Sandhya, over to you.
Thank you, Umra. Good afternoon, everyone. I am Sandhya Yadav. I am proud to tell you that I am a second year MSc internship student and a member of the webinars organizing committee here at QPS University. It is truly an honor to serve as the moderator for today's enlightening webinar. We are privileged to have with us Professor Dr. Kumar, a globally respected expert in environmental microbiology, industrial microbiology, environmental friendly products and technology, and polymers. He is associated with the School of Biological Science. Professor Sudesh holds a PhD in molecular biology from Saitama University, Japan, and is widely recognized for his interdisciplinary research connecting ecology, society, and culture. His scholarly work bridges the biodiversity and societal needs, particularly in the areas of bioculture, biodiversity, environmental microbiology. He brings not just academic experience, but it also a unique perspective on how science can serve both nature and communities. His voice is one of wisdom, awareness, and action. Today, he will be sharing his insight on the topic biotechnology for a sustainable future, innovation in biological production. With his brief of Professor Dr. K. Sudesh Kumar to deliver his talk. Professor Sudesh, over to you.
Okay, thank you very much, Ms. Sandhya, and then Ms. Umra, Ms. Priyanshi, and also to Prof. Subhash for inviting me today for this BioConnect webinar series that is for students, by students. I think it is a very nice initiative. So now let me try to share my my slides. Hopefully it goes well. And then I should. Okay. So I hope my slide is now clear and you can also hear me clearly. Is everything okay?
Yes, yes, yes. Please go ahead, Professor
Alright.
Sudesh.
So thank you very much again for inviting me to this webinar series. My name is Sudesh, and I'm from Universiti Sains, Malaysia. And I will be talking about biodegradable plastics. So biodegradable plastics is one kind of bioplastic. So I will try to take you through the introduction about bioplastics, and then what is the difference between bioplastic and conventional plastics, and then how do we synthesize bioplastics, and what are the challenges, and also finally what kind of research that is needed in order to further commercialize this bioplastic. So the title of my presentation is Biotechnology for a Sustainable Future and Innovations in Bioplastic Production. So the first slide, as you can see, there are many different items on the slide. On the left in the circle is the microorganism that is used in this production of bioplastic. And this is an image that you get if you view the bacterial cells using a phase contrast light microscope. So I don't know if it's clear for you to see the small rod-shaped structures that you see are the bacterial cells. So in this picture here, probably there are thousands of bacterial cells. And to grow these bacterial cells, you need various components. I think what probably you can see clearly is a cylindrical object, stainless steel cylindrical object, which is the bioreactor. And this is just a very small bioreactor. It's only about 150 liters in size. The actual commercial bioreactors are probably 10 times or even 100 times bigger than this. So this is still, it's a small pilot scale. And then you probably also see a bottle of oil, near the bioreactor. So in my project, we are focusing on using vegetable oil as the carbon source, as the feedstock to feed the bacterial cells in order for them to produce the bioplastics. And then you also see some other items, and I think maybe you are attracted to a creature that looks like a worm that is there. So probably you are wondering, what is this worm doing here in this bioplastics topic? So I will try to introduce you to the role of this worm in this whole process. Okay, and I would like to keep this presentation as informal as possible. So try to ask, anything if you feel like you want to ask about some things that you are not so clear, then you can ask even during the presentation. So first, I think I should briefly introduce to you about my location. So I'm in Malaysia. I think you know Malaysia, which is not too far away from India. And my university is in a small island called Penang, which is in the northern part of the peninsular Malaysia. It's quite close to Thailand, actually. And lately, I think there are direct flights from India to Penang. So it is actually quite easy. And I think it only takes about 3 hours, if I'm not mistaken, for the flight. Okay, so it's not too far. So I hope some of you all will be interested to come to Penang and to visit my university. And Penang is a very famous island for tourism. There are nice, beautiful beaches. And Penang is a place where there are many different races, like Chinese, Indians, and Malays. And also, of course, you can imagine the food is also quite diverse. So this is one kind of food that is famous in Penang. Of course, there are more. So I hope if you come to Penang, you can enjoy not only the views and also the food here. So let me now go into the issue or the topic of this presentation, which is plastics. I think all of us are very familiar and we know what plastics are. And there are many different kinds of plastics that we see in our daily life. So this picture here shows some of the uh, things that we use in our daily lives, which are made from plastics. So we are very dependent on plastic items, and it's not easy for us to live without plastics. The last two pictures on the right are basically labels from clothes. So plastics are not just items that we use. That is like those pictures that you see on the left. But our clothes are also made out of plastics. I don't know if you realize that. If you look at the label, you will see that there are components like polyesters, nylons, and polyacrylic, and all these are basically different types of plastics. So plastics are everywhere. It is close to us, and it is also uh, in various components of products that we use in our daily life. Recently, there was a paper published. This was last year, addressing or highlighting the issue of microplastics. And I don't know if you have heard of microplastics or nanoplastics. So these are small plastic debris or particles that are generated from all the kind of plastics that we use in our daily life. So plastics, when they decompose or when they are subjected to various physical forces like friction and erosion, they break down into smaller particles. So this results in microplastics. And these microplastics, because they are not biodegradable, they remain in our environment for a very long period of time. So this paper, which was published last year, highlights the quantity of these microplastics that are being consumed by people all over the world. And what was interesting is that Malaysia was highlighted as one of the top countries which the population consumes, of course, not intentionally, large quantities of microplastics, which get into the food supply through the food that we eat. So this includes uh, aquaculture products. I mean, aquaculture, um like fish and shrimps, and also from the ocean. And not only that, even from the atmosphere, from the air, large quantities of microplastics are being inhaled into our body on a daily basis. So this is quite alarming because it highlights the fact that we are consuming large quantities of microplastics, and the effect of these microplastics are beginning to show in our health and in our well-being also. So I think this kind of problem is not only limited to Malaysia or to Southeast Asia, but it is global. It is everywhere. So it is a serious issue that needs to be addressed as soon as possible. And besides microplastics, there are also single-use plastics that are used only once, like in the case of plastic packaging or plastic bottles, that are used and then thrown even immediately after the contents are used or consumed. So there are mounting waste of single-use plastics accumulating all over the world. So this pictures showing, the amount of single-use plastics after use that are discarded. So they are not only on land, but also in the sea, in the river, and almost all ecosystems. So this is really alarming. So I think we all understand that plastics are useful and we need to use plastics for a comfortable lifestyle. But the problem is after use. What do we do with the plastics that are so good that it doesn't get away or it doesn't decompose easily? Okay. So now let me introduce to you about bioplastics and also the conventional plastics. So we all know that plastics, the conventional plastics are from petroleum. Okay, fossil fuels are used in order to get the building blocks to polymerize and to synthesize plastics. So these are the conventional plastics like polyethylene, polyethylene terephthalate, polypropylene, and all. So this is shown in the quadrant on the top left, which is in the black circle. So these conventional petroleum-based plastics, they are not bio-based. Bio-based means those type of material that are renewable, which means that they are produced from vegetable matters or vegetable oil or something that is renewable. And petroleum is not a renewable resource. Okay, so this is not bio-based. And then the conventional plastics that we are currently using are also not biodegradable. So that is why we have all these problems that I've already shown in the previous two slides. So nearly all types of conventional plastics that currently we use are not bio-based and not biodegradable. And then if you look at the quadrant on the top right, you also have not bio-based plastics, but biodegradable, which you can also produce from fossil or petroleum resources, okay, which is not bio-based. So it is possible to produce biodegradable plastics from petroleum. So this is where you have plastics like polybutylene succinate. It is abbreviated as PBS, and then polycaprolactone, PCL. So these are all plastics that are not bio-based. That means they are from petroleum, but these are also biodegradable. So it is important to know that you can produce biodegradable plastics from petroleum. Okay, and then if you look at the quadrant on the bottom left, so then you have bio-based but not biodegradable. So this is also possible, okay, due to scientific research and development. So now it is possible to produce plastics just like petrochemical plastics like polyethylene and polyethylene terephthalate, but the monomers actually comes from biological processes. So this is where you have some confusion and complications when you use the term bioplastics. So technically, biopolyethylene and biopolyethylene terephthalate are bioplastics because the monomers are produced from biological processes or renewable substrates. Okay, so you have here bio-based but not biodegradable. Okay, and then if I go back to the quadrant on the top right, you have not bio-based but biodegradable. So this is also a kind of bioplastic. So if somebody asks you, is polybutylene succinate, is it a bioplastic? The answer is yes. Polycaprolactone, also the answer is yes. Okay, although they are produced from petroleum, but these are bioplastics. Okay, so I think you can see the complication or the confusion that can arise. But the one that I'm going to talk to you all more in this presentation is a kind of plastic that is biodegradable and bio-based. Okay, so this is not from petroleum resources. So these are from renewable resources like vegetable oil. and they are completely biodegradable. So this is where you have plastics like a PHA. This is in short for polyhydroxyalkanoate. It's a very long scientific name. So in short, I will call it as PHA. And then you also have polylactic acid, which is also another kind of bioplastic. And then you have starch, okay, and starch blends, which is also a kind of bio-based and biodegradable plastic. Okay, so this is a little bit confusing, and you may want to later look back at this slide and try to understand, so that you can see that there are many kinds of bioplastics. So sometimes you see in newspapers or in television, people mentioning bioplastics and then saying bioplastics are not biodegradable, and then bioplastics are not environmentally friendly. So the confusion is because people are using a very general term to refer to many different kinds of bioplastics. But the bioplastics that are biodegradable and environmentally friendly are the kind of bioplastic that is shown in the quadrant on the bottom right, okay, which includes PHA, PLA, and starch-based bioplastics. And there are already, a lot of companies that are producing many kinds of bio-based and biodegradable plastics. And this is because of the problems, okay, that I've already highlighted in the previous slides. So PHA is the one that I'm going to talk to you all more. So there are already also a lot of companies from different countries that are producing PHA and already making products from PHA. So these are some examples of PHA-based thermoplastic products. So thermoplastic is probably a new terminology here, which means these are materials that can be thermally processed, which means by using heat, you can melt this material and then mold or shape the material into certain shapes, and then it can be hardened. So, and this process can be repeated. So you can melt the material again and remold it. So this is what thermoplastic means. So there is another term which is quite similar, which is thermoset. So thermoset is different from thermoplastic. Thermoset, you can only mold it once, and then it cannot be recycled back to the original form. Okay, you cannot melt a thermoset and make it into a different product. But thermoplastics, yes, you can melt and then can reprocess and recycle. Okay, so these are some examples of products that are made from PHA-based thermoplastic. So you can basically make any kinds of plastic products using PHA. So now let me introduce to you about PHA, or polyhydroxyalkanoates. So PHA is a polymer or plastic that is made entirely by bacteria. Okay, I think you all know what is bacteria. So bacteria are small organisms. They are microorganisms that are present everywhere. They are present in the soil. They are present also in our body, in our intestine. They are present in water, everywhere. Okay? So microbes are basically involved in many ecological processes. And some of these bacteria, they have the ability to store this kind of polymers. So polymers are basically all kinds of large macromolecules which have various functions. So usually, like even our body, if you look at the components like protein is also a polymer. So protein is a polymer made of amino acids. Okay, and then besides protein, you also have carbohydrates. Okay, carbohydrates is a polysaccharide, which the monomers are sugars. So for example, glucose can be polymerized to make starch. Okay, so likewise, microorganisms, they make polymers in order to store their carbon sources or their food. Okay, so polyhydroxyalkanoids is actually a food storage compound that are synthesized by many kinds of bacteria. So for them to survive when there is no food from the environment. So that is why bacteria makes this material called PHA. So they are not making plastic for us. Okay, they are making a kind of polymer that they can store inside their cell, which has properties just like the plastic that we use. Okay, so how does this bacteria make this kind of plastics? Is you have to feed them with large quantities of carbon source, okay, food that they will store in their cell cytoplasm. Okay, so this storage compound, which is the result of feeding the bacterial cell with large quantities of carbon source like oil or sugars, is what you get in the form of granules, PHA granules. So all these white spherical objects that you see inside this bacteria, it's a cartoon, is the PHA granules. And the key enzyme that is involved in the synthesis of this PHA granule is the PHA synthase enzyme, okay, which is encoded by the PHA synthase gene. Okay, so this protein structure is shown in this protein model, okay, which is rotating, as you can see. So this is the structure of the protein, okay, the PHA synthase, which is involved in synthesizing or making this PHA. And for this, you need to feed the bacteria with large quantities of feed that they can consume. So the kind of feedstock that I have been experimenting with in here in my lab are mostly derived from oil palm plantations. Okay, as you may know that Malaysia and Indonesia are two of the world's largest producers of palm oil. I don't know if you are familiar with palm oil or whether you use palm oil in your place. But in Malaysia and Indonesia, palm oil is widely used, okay, for cooking. Okay, and the process of getting palm oil from the oil palm plantation results in various byproducts. Okay, so these are the waste that you can get from the processing of palm oil, which we can use to feed to the bacterial cells. So we try to use waste products and also those that are cheap, okay, in order to produce this PHA. And the reason is because we want to reduce the cost of making PHA. Okay, and then comes the recovery. So because this PHA is an intracellular product, which means that this PHA is produced inside the bacterial cell and it is stored inside the bacterial cell. So in order to use it, you have to extract or recover these PHA granules from the bacterial cells. So this is not an easy task. Okay, it's not easy to break the bacterial cell and then to extract the polymer, the PHA granules, and then you also have to purify the PHA granules from other cellular compounds, okay, like lipids or proteins or all kinds of other components. So this is quite challenging, right? So once you have purified the polymer or the PHA granules from the bacterial cells, then what you get is something that looks like starch, okay, which looks like a white powder, just like starch. Okay, but these are actually the PHA granules from bacterial cells. Okay, and the type of PHA that we are producing right now is a copolymer, which is made out of two different types of monomers. Okay, there is a 3-hydroxybutyrate monomer, which is a 4-carbon compound, which is copolymerized with a 6-carbon monomer, okay, which is 3-hydroxyhexanol. So the reason why these two different monomers are used is to be able to tailor the properties of the material. So just like the kind of plastics that we normally use, you will see that some plastics are soft, some plastics are hard. Okay, like if you take a food container, the container can be hard, but the cover, the top layer can be soft. Okay, so in order to make all these different types of plastics, you need to be able to control their molecular structure. So this is where two different monomers is it allow us to control the properties. So later I will show you how we can control the properties of the material by controlling the molecular structure of this material. So you are probably wondering, where does that worm-like structure comes in? So the worm-like structure comes in here in the recovery process. So this is something that we discovered, okay, which um, makes the recovery process much easier. Okay, so we are using the worm, which is a kind of mealworm. I don't know if you have seen mealworms. So those of you, if you are having pets at home, like birds or some some reptiles and all, so you are familiar with this mealworm. So mealworm you can buy from pet shops. Okay, and this mealworm can be used to recover the PHA granules from the bacterial cells. So later I will show you how we do this. So palm oil, I think I don't need to introduce too much. So it is oil that you get from oil palm trees. So this is how an oil palm tree looks like. And at the top of this tree, you have bunches of oil palm fruits like this. Okay, and then the fruits, if you take a cross section of the fruit, you will see that it has a kernel, just like coconut. It has a fibrous outer layer and a shell in the middle, okay, the kernel. But unlike coconut, which is big, these are small. These are small fruits, but they form in bunch. So each of these fruit bunch can have about 2,000 fruits. So the oil that we get from this oil palm, there are two types. One is the oil from the mesocarp, the flesh of this fruit, which is used for cooking and for all kinds of vegetable oil. And the kernel oil, which is called palm kernel oil, is used as a source of oleochemicals. Okay, so these are the two main sources, usage of palm oil, okay, as food and also as a source of oleochemicals. And then, of course, now because of the demand for biofuel, the oil can also be feedstock for the production of biofuel. And in my lab, we have been focusing on using the waste from the palm oil industry to use it as a carbon source, as a feedstock to feed bacteria to make bioplastic. And you may be wondering, or you may be asking, why are we using palm oil? Okay, the reason is palm oil is the most productive oil. The graph here shows that the amount of oil you can get per year in an hectare of land. So you can see that the palm oil is the most productive. It means you can get the most amount of oil in an area in a year, okay, compared to all other vegetable oils. So it is something that is very productive, and it is also, because of that, it is cheap. Therefore, it is a interesting feedstock that can be used for making all kinds of biotechnological products. So this is what I was telling you earlier, that we are using the waste from the palm oil industry. So when the oil is processed, the waste that comes out can be recovered and used as a feedstock to make PHA. Okay, later I will go into more details here. I'll move through. And of course, palm oil is not available in all countries. Okay, I don't think palm oil is produced in India. Although I think Malaysia exports quite a lot of palm oil to India and also to other countries. So not all countries have palm oil. So, for example, other countries may have other types of oil, okay, which you can also explore to see if they can be used as a feedstock to produce PHA. So, over the past few years, we have collaborated with many researchers from other countries. So, this research result is in collaboration with Nigeria and also China. Okay, so here what we did was we tried to use other types of underutilized plant oils, okay, like desert date kernels, bitter apple seeds, and African elemi fruits. So these are all oils that you can get from these types of seeds, but they are underutilized. They are not utilized for for high value application. So we did this study, and then we found that yeah, these types of oils are also consumed by the bacteria, and the bacteria can produce bioplastics. So I think likewise in India also, we did a study before using mahua oil. Are you familiar? Maybe you have heard of mahua flower. So the mahua seed also contains high amount of oil, which can be used to produce this type of bioplastics, PHA bioplastics. And we also had a collaboration with Vietnam. So in Vietnam, they don't have plant oils as much, but they have this waste fish oil, because fish oil extraction is a big industry in Vietnam. And in the process of extracting valuable fish oil, you get the waste fish oil, okay, which are not utilized. So when we carried out this study together with researchers in Vietnam, we found that waste fish oil is also a very efficient feedstock that can be fed to certain types of bacteria to produce PHA. And similarly, we also did a research collaboration with Oman. And you probably know Oman is a country that produces a lot of dates. And dates contains a seed, a kernel, which also contains about 10 weight percent oil. So we extracted the oil from this date seed. So the seeds are normally not used, they are just discarded. And the oil in the seed can be extracted, and we tested for its ability to be as a carbon source for bacteria. And then we found that bacteria can use the seed oil and make PHA. So this is also something very interesting. So if you want to produce PHA in India, I think you have to look for a kind of feedstock that is cheap and widely available that can be fed to bacteria to produce PHA. And we also collaborated with our research collaborators in Germany. So these are some of the researchers who came over to our university, and then we carried out PHA production using low quality animal fats. So animal fat is also an interesting feedstock, okay, that is a waste that you get during processing of animals. And these fats can also be converted into feedstock that can be used to produce PHA. So what I'm trying to show to you is that there are many different kinds of feedstocks that can be used in order to produce PHA. I think next is a video. I hope you can see this video. So this is a process of how bacteria is cultivated to produce PHA. So this is shake flask to produce the inoculum. Then it is scaled up to the fermenter, and the cells are harvested. And we use drum drying to dry the cells. So it's a very short, quick video, but basically it shows the entire process of culturing the bacteria from inoculum building up to the fermenter, and then after growing the bacteria in large scale, it is then harvested and subjected to recovery. So the recovery uses mealworms. So the mealworms are able to eat the bacterial cells, but they are not able to digest the PHA, the polymer. So therefore, all the PHA comes out in their excrement, in their feces, so which can be recovered and washed and easily purified. So we are currently in the process of scaling up this entire process from the shake flask to 13-liter bioreactor, which is then further scaled up to 150-liter bioreactor, 750-liter bioreactor, and finally 2,000-liter bioreactor. So 2,000-liter bioreactor is still small. It is still a pilot scale. The industrial scale would be like 10 times this size, about 20,000 liters. Okay, so we are now in the 2,000-liter scale where we can produce PHA like a few hundred kilos every month. So now let me, I think I have, please let me know if my time is up, so I can introduce to you a little bit about the type of properties that you can get by controlling the monomer composition. So if you still remember just now, I showed that the type of PHA that we are producing now contains two different monomers. Okay, the smaller one, which is 4 carbon, which is called 3-hydroxybutyrate, and then the one on the right is 6 carbon, which is 3-hydroxyhexanoate. So by controlling the ratio of these two monomers, we can control the properties of the PHA. So for example, when we increase the 3-HHX, the HHX fraction from 0 to 30 mole %, okay, so we can control all these properties. For example, the melting temperature of the material. So by increasing the HHX molar fraction, we can decrease the melting temperature. So this polymer can be melted at a lower temperature. And by increasing the HHX molar fraction, we can also control the flexibility of the polymer. So in other words, higher HHX gives you a polymer that is more flexible. The crystallinity is also an important feature of materials. The more crystalline a material is, the more harder it becomes. So the more amorphous is the opposite for crystalline. So making it more amorphous makes it more flexible also. And the last characteristic that we can control is the opacity, meaning the transparency of the material. So if you can see the picture on the left, so the cap of this container is transparent. So you can see my fingers behind the cap. Whereas the container on my other palm is not transparent. So you can't see my fingers, okay, underneath that container. So this is the different properties that we can control by controlling the monomer composition or the molar fraction of two different monomers. So with this, now we can make different kinds of products, okay, like which is shown here, like fork and spoon and small containers. These are just examples of products, to show that it is possible to make almost any kinds of plastic products using PHA. Okay, so these are further examples. Okay, and I've actually shown and presented these products in other places also. So this is in one university in China. So where the researcher there wanted to check the thermal stability of PHA spoon. So what this researcher did was to pour hot water, which is close to about 100 degrees, and to see whether the plastic is still strong. Okay, because some plastics are not thermally stable, and when it is put in hot water, it melts or it becomes soft, and it's not suitable for hot conditions. But PHA is very thermally stable, which means that you can use PHA even in close to 100 degrees Celsius. So next is biodegradation. I think you want to see evidence that this plastic that comes from bacteria is biodegradable. Okay, so what we did was to make different kinds of samples like films and bury the films in soil and then see how long it takes for it to completely biodegrade. So here are various types of PHA, the P3HB, PHBHX, and all are different types. So all types of PHA materials are 100% biodegradable. So all of these samples, they degraded completely within 3 months. Okay. And then here are further products that were made and then which were buried in soil and then which were put in seawater, fresh water, and within like 4 months, you can see that a significant portion of it has been degraded. Okay, here are further examples like straws, which are also degraded like within 6 weeks. Okay, some further explanation about the mealworm process of recovering the polymer. So as you can see here, the mealworms, they can eat the bacterial cells, and then the feces or the excrement of these mealworms will be white in color because the polymer is actually white. And when we observe it under electron microscope, you can see that the PHA granules mostly the feces are composed of PHA granules. So this process is eco-friendly. It doesn't use chemicals or solvents. So, and it is also like getting two products out of this process. So one is the mealworm, which can be used as a feed for animals or for pets. And then you also get the PHA, as a second product. So mealworms are also gaining a lot of interest as a source of protein, because insect protein is more eco-friendly compared to animal proteins, and it is also more sustainable. So mealworm is a larvae of a beetle, and this larvae has got very high protein content. So you can see me, the picture on the left is myself, and the picture on the right is one of my collaborators from Sweden. So we attended this world's first conference on insects for feed and food in the Netherlands in 2014. This is almost 10 years ago. So at this conference, they gave many kinds of food during tea breaks that contain insects. So this is real insects, okay, that we are eating, to show that this insect protein is gaining popularity. So why are we using mealworm or insects to recover the PHA? Is because we want to bypass this expensive and very instrument based recovery process. So the PHA granules, in order to recover from the bacterial cells, has to go through various process as shown in this flow chart. So by using mealworms, we can cut through, okay, we can bypass all this process and make it more cheap and also eco-friendly. Okay, that's the reason why we are using mealworms. Okay, so, and this is my, I think, final conclusion slide, okay, to show that we can use various kinds of renewable resources and also waste that we get from our other processes like plants and animals, which can be used as a feedstock for making PHA. So this is important not only for sustainability reason, but also to make it more economical. So one of the reasons why PHAs are not yet commercially available in all countries is because it is expensive. So we hope by using this cheap feedstock and also by using this mealworm process to recover and purify the polymer, we can reduce the cost further. And it is important to grow the cells at high cell density so that we can reduce the cost of making PHA. And also the biological recovery of PHA using mealworms can also further reduce the cost. And in the process, you can also get insect proteins, okay, as a secondary product. And in the process of producing, all these kind of eco-friendly products, I think it is important to bear in mind that we don't want to contribute to loss of tropical rainforests and biodiversity. We don't want to cause problems to animals' habitat or loss in, this kind of habitats. And global warming and climate change, I think we already feel in Malaysia now the climate is getting hotter. I don't know about India. I think India also should be the same. And finally, conversion of food to fuel or material is something that we don't want to contribute to because food is also important. Food is also necessary. So we don't want to convert food grade material into non-food compounds like plastics. I think these are all important issues that we have to keep in mind when we develop biotechnological products. Okay, so with that, I would like to thank you and I would be happy to answer any questions that you may have. So thank you very much.
Thank you so much, Professor Sudesh, for such a rich, insightful, and thought-provoking session. Your words have depended your understanding of bioplastic production, not just a scientific concept, but as a vital relationship with nature that we must foster and protect. Now we will move into the question and answer session. I encourage all participants to keep your question ready. You may type them into the chat box. We will do your best to address as many as possible within the time we have. To begin, I would like to pose a few questions to your respected speaker. Professor Sudesh, what are the most promising source or raw materials for sustainable bioplastic production today?
Okay, thank you for the question. I think this depends on where you are. So for example, like in Malaysia, the most promising feedstock is of course palm oil because palm oil is produced in large quantities. But if you're in India, of course, palm oil is not interesting feedstock. So you probably have to look at other feedstock. I think maybe sugarcane industry may be quite important and big in India, or maybe rice or wheat. So the products that you get from the processing of this agricultural commodity may be interesting feedstock for making PHA.
Thank you, sir. Now I proceed to another question. How do bioplastic compare with conventional plastic in terms of performance and cost effectiveness in industrial application?
So cost effectiveness, of course, plastics are much cheaper. Conventional plastics are very, very cheap. That's why it is widely used in all kinds of applications. And bioplastics, as you can see in my presentation, it is very difficult to produce. So first you need infrastructure like fermenters, and then you need to grow the microbes, and then you have to feed the microbes with carbon sources. So there is a lot of cost involved in producing PHA. Okay, so we cannot compete with commercial like petrochemical plastics. Petrochemical plastics are too cheap. So it is impossible to get bioplastics at the price of petrochemical plastics. But if you take into consideration the end of life cost, so after plastics are used and then they are discarded, the waste treatment cost of plastics, if you include them into the cost of plastics, then maybe bioplastics may be almost the same as the cost of petrochemical plastics.
Thank you, sir. Now I proceed to another question. Could you elaborate on the environmental impact of bioplastics? Are they truly biodegradable under natural conditions, or do they require industrial composting?
Yeah, that's a good question. Not all bioplastics degrade naturally in the environment. PHA, because they are produced by bacteria and it is a food storage compound for bacteria, uh, because of that, most microorganisms in nature, they have enzymes to degrade PHA because it is a source of food for bacteria. But there are other bioplastics which are not uh, a source of food for bacteria. So they naturally don't have the enzyme for decomposing or to break down bioplastics. So for example, polylactic acid. Okay, polylactic acid is also a bioplastic. It is also bio-based, but it is not biodegradable. The reason is because bacteria naturally they don't have enzymes to break down polylactic acid. So you need composting. So composting, what really happens is the high temperature that is generated during composting. So in compost, normally the temperature rises up to like 60 degrees or close to 70 degrees. So at this temperature, there can be thermal decomposition of some materials. So like PLA, they are thermally decomposed by the high temperature in compost, and then the smaller compounds are then degraded by bacteria. So that is why you need composting for some kind of bioplastic. But for PHA, not necessary. PHA, they can, of course, break down or biodegrade in compost, but PHA can also biodegrade naturally in all ecosystem as long as there are microorganisms. Okay, I
Thank
hope
you, sir.
my answer is. Yeah, any other questions?
Yes, sir. What are the current technological challenges in scaling up bioproduction globally?
Yeah, the challenges in scaling up PHA production is the availability of feedstock. So as I mentioned just now, we can use waste from various agricultural products. Okay, for example, oil or sugars and all. But the amount that is available is quite limited. So plastics currently, the world production of plastic is about 400 million metric tons per year. So that is a very huge quantity. So in order to produce that amount of material like PHA, I don't think there is sufficient feedstock. So we don't have enough feedstock to produce such large quantities of bioplastic. So that is one of the challenges.
Thank you, sir. Now I warmly invite to your participant to unmute yourself if you like to directly ask your question to your esteemed guest and fellow student. That has indeed remarkable session. I take this opportunity to extend my heartfelt thanks to Professor Dr. Kumar for his valuable time and inspiring insight. I also wish to thank all participants for your active involvement and curiosity. Now I hand it back to your MC, Miss Umra. Over to you. Thank you.
Thank you, Santhya, for effectively moderating the session. Distinguished guest and fellow student, as we approach the end of today's inspiring session, I now invite Ms. Neha Khan to deliver the vote of thanks on behalf of the organizing committee. Neha, over to you.
A very warm good evening to everyone as we draw this insightful session of the BioConnect webinar series to a close. It is my privilege on behalf of the entire CSRD student team at People's University to deliver the vote of thanks. We begin by expressing our heartfelt gratitude to our Honorable Vice Chancellor Professor Dr. Harish Rao for taking the time out of his busy schedule to deliver opening remarks for this program. A special note of thanks to our distinguished guest speaker, Professor Dr. K. Sudesh Kumar, for delivering a thought-provoking talk on biotechnology for a sustainable future, innovation in bioplastic production. Sir, your insights have broadened our academic perspective and ignited our curiosity to explore real-world solutions through biotechnology. We extend our deep appreciation to our mentor and guiding light professor, Dr. Subhash J. Bhore, head of CSRD, for his unwavering support and encouragement of student-led initiative like this one. Sir, your vision and leadership continue to inspire us to aim higher. A heartfelt thank you goes to our moderator, Ms. Sandhya Yadav, for smoothly steering the session and encouraging meaningful dialogue. We also sincerely thank our organizing committee chairperson, Ms. Priyanshi Sharma, the committee member, and the entire CSRD student team. Your dedication and behind the scenes effort have been instrumental in making today's event a success. To all our enthusiastic participants and attendees, thank you. Your presence and engagement bring life to this platform and motivate us to keep growing and learning together. This is just the beginning. The BioConnect webinar series is envisioned as a vibrant student-led platform where ideas ignite innovation, where students evolve into leaders, and where knowledge translates into meaningful action. We hope today's session was as enriching for you as it was for us. And we look forward to welcoming you to future chapters of the BioConnect webinar series for the student by the student. Before we formally conclude, we would like to offer our special thanks to Shri Suresh and Vijayvargi, Honorable Chancellor of People's University, and Dr. Megha Vijayvargi, Honorable Trustee of Pro-Chancellor, People's University, for their visionary leadership, constant encouragement, and continued support, which remain the cornerstone of academic excellence at our university. May your journey be filled with curiosity, innovation, and meaningful discoveries. Wishing you all a pleasant evening. Now I will hand it back to our MC, Ms. Umra. Ms. Umra, over to you. Thank you. Over.
Thank you, Neha. Once again, thank you to everyone who have joined us today, especially our speaker, vice chancellor, mentor, and participants. It was a great session today. Before ending this session, we would like to hear feedback from all participants. This post-webinar survey link is posted in the chat box. We promise the evolution survey will not take you more than 2 minutes. Your feedback will be greatly appreciated. We will want for a few minutes to let our participants complete their feedback form. I hope that everyone has given their feedback. Please note that you will receive your e-certificate shortly. We look forward to seeing you in our next BioConnect session very soon. Until then, stay curious, stay inspired, and keep connecting. This is Umra signing off on behalf of the CSRD student team at People's University. Have a wonderful evening.